inviting me for the one-day international webinar. Thank you. Good morning to everyone. Am I audible? My voice is clear, no? Okay. So I will share my PPT. Can you see my screen?
It's visible only, sir.
It's okay. Is it okay, ma'am?
Yes, sir.
Yeah.
Sir.
Yes, ma'am. Good morning, my dear students, nursing students.
Morning, sir.
Good morning. Welcome you all. Thank you very much to give me the 1 hour slot to discuss about my research work. And currently, it's a big issue, especially for women and as well as in men also. So today's the topic is genotypic distribution and molecular epidemiology of human papilloma virus. Shortly we call HPV in pap smear samples. What is pap smear samples? It's a cervical sample. Just scratch or scrap the cervix of the female, and then we can get some loose tissues, free tissues, that is called pap smear samples of women in the United Arab Emirates using real-time PCR study. So, yeah. So you know very well about cancer. All of you know about the cancer, especially men is prostate cancer, lung cancer, pancreatic cancer, liver cancer, and gastric cancer, stomach cancer, all are there. In female also it is there, same. Lung cancer is quite common in female, but which one is more predominant? There are two cancers are more predominant. One is breast cancer, another one is cervical cancer. You can see here the cervical cancer. The cervical cancer worldwide is the second most common cancer among women throughout the world. According to the WHO, they have mentioned that the cervical cancer is the second most common cancer. So fourth most common cancer related death among women globally. So the WHO, they have recently announced that, stated that almost 660,000 new cases of cervical cancers are progressing. They are identifying. So new cases they are identifying, and almost 350,000 deaths caused by the cervical cancer. Especially they identify in the low and middle income countries. What about the high income countries and developed countries? They have more awareness. But in the low income countries and middle income countries where the government need more improvement, or where the people should know more about the cancer, the awareness program. So government doesn't failure to give more awareness related to the cancer. Now entire worldwide, every person, they know about hepatitis B virus, HIV, mainly HIV. What about the cancer awareness for female breast cancer or cervical uterine cancer? They know, but they don't know the real actual the data about the cervical cancer death, especially in low income countries and middle income countries. So, and another one is, what is the specialty about this cervical cancer? Third most frequent cancer among the women, the age between 15 to 45 years. So it is one of in UAE, it is one of the 13th leading cause of cancer. So first one, second one, third one, like that. They have categorized, they have hierarchy like that. So in the cervical cancer related mortality death is a 13th position. So, okay, we are discussing about the cervical cancer. What causes or which agent causes the cancer, the cervical cancer? The predominant one is a human papilloma virus. This human papilloma virus transmitted through the contact. So through the sexual mode or maybe skin contact, like that. But usually through the sexual mode, it enter into the cervical area and causes cancer. Possibilities are there. So the human papilloma virus, nearly more than 450 genotypes, subtypes, human papilloma virus genotypes, they have classified. Now, today, as of now, more than 450 genotypes of human papilloma virus have been identified by many scientists. So, approximately among the 450 genotypes, approximately 40, nearly 40, capable of transmitted through sexual mode, especially anogenital tract. Many of them among the 40, they are very high risk. They are very dangerous to develop cervical cancer. So what is that HPV? The HPV is a virus, you know very well. I think you have studied in the undergraduate. Yeah, you are now in the undergraduate, either maybe studied in first year or second year, virology. Maybe if you are not studied, maybe you will study later, especially nursing student. So I think you have the paper microbiology and pathology. So anyhow, you will study this virus and as well as pathological changes of cervix and something like that. This is the virus. You can see this is the HPV virus. It's a DNA based virus. Viruses have been classified into two groups. One is a DNA viruses, another one is RNA viruses. This human papilloma virus is a DNA viruses. It's a simple. They have capsid and then DNA. The DNA is a nearly 80 kilobase per in size, very small size. This genome is synthesizing nearly 9 proteins, envelope protein, early protein, that is E1, E2, E3, E4, E5, like up to E7 proteins, they have synthesized. Along with that, late protein. That late protein is the envelope protein, L1 and L2. These two proteins are synthesized. So total 9 proteins the viral particle will synthesize. And then go for the replication. So these proteins are very important. Out of these, among these 9 proteins, E6 and 7, they are very important to cause the cancer. So according to the classification nomenclature, the HPV genotypes have been again subgrouped into two. One is a high risk HPV genotypes, and another one is a low risk HPV genotypes. What is high risk? High risk and what is a low risk? So if someone get the high risk genotype, for example, 16, sure, it will develop the cancer. Not now, maybe after 5 years or 6 years or maybe 10 years, it will take time. But depends on the body condition of the person, those who have the HPV-16 or 31, 18 like that. Nearly 20 genotypes we have to go for, we have to identify, we have to go for the test. So these high risk human papilloma viruses, they are grouped into high risk level. They are prone to cause cancer. Low risk, it will develop, but it will come very little relax, or maybe the progression of the cancer will be low risk, isn't it? So maybe benign or may not, maybe or may not be. That is called the low risk genotypes. So nearly 12 genotypes low risk. Total 32 genotypes we have identified, but many research papers and the latest one, they have identified 40, but among the throughout the world, more prevalent, these 32 genotypes are more prevalent. So if someone infected by either 16 or 6 or 18 or 11 is a genotype, if it is persist, may develop cancer, but a high risk is sure it will cause the cancer. What type of cancer? Invasive cancer. That means the cancer cell invert between the normal cell and then spread all over the body. Metastasis will form. So now in UAE, and the crude incident rate, so we don't have the exact rate how many people died due to the HPV related cancer. So now we have consolidated crude data, 4% per 100,000 population. So we need actually our study group, our study team, research team, we need more clear data available on how many people died due to the HPV cancer. Among that, population, how many are infected by low risk HPV, high risk HPV. So this burden we should know among the among the UAE study population. Then only we can go for further step. I will let you know in the end of the slide. You will come to know why I'm talking about high risk, low risk. Then what is the relationship between the high risk, low risk with the population, study population, especially female, and then why the government should take further steps to reduce the burden. You will come to know the after 1 or 10 slide. Maybe you will understand. Oh, this is the main purpose. That's why the study is focusing on HPV genotype distribution and molecular epidemiology. That's the thing. But in India, the floating populations we are all Indians, we are living in the India. But in UAE, multi-nationalities are there. So maybe this year many countries will come, and the next year another country will come, like that. But existing populations are certain UAE nationalities, and then certain Indians, and then Asian people, they are permanent. But other people, they come and go like that. What will happen? They can spread the viral agent to the those who are living in this country, isn't it? So permanently, those who are residing here for permanently more than 15 years, 13 years, 30 years or something, they may acquire the human papilloma viral genotypes from the floating population. So next, I will show to you. I want to show to you. This is a human papilloma viral infection. After the infection, what are the changes will occur in the cervix area? So if the viral particle enter and multiply, you can see different changes will be there. This is the pathological identification. We have to collect the cervix, the free floating, free available cervix. If you scratch or scrap the cervix area by using brush, so the free superficial tissue cells will be collected. That cells we can stain and then we can identify. That means that the squamous epithelium is the main layer. I will show it to you here. Yeah, this is the one. Then I will come to the next previous slide. This is the normal cervix. When you check in the normal healthy woman, this is the normal cervix. This cervix is this is the layer, normal cervix structure. Squamous epithelium, this is the squamous epithelium. The superficial layer, this is the superficial zone, middle zone, basal layer. Then you can see the violet color. This is a basement membrane. Then dermis will start. This is the thick layer is there. So if the viral particle enter here, first what they will do, they will attach, infect the basal cell. So if the viral particle enter here, first what they will do, they will attach, infect the basal cell. This cell, it will be there. Then viral particle will replicate. After the replication, the DNA, HPV DNA will amplify within the cell and try to go for viral assemble and multiply, viral progression, and then each cell will be infected by the viruses. Then how long the viral will be persist in this cell? Maybe decades, maybe 10 years, 15 years, 20 years, like that it will be persist in the infected patient cells. So once it infected, some changes will be there. What is the changes? LSIL stage, HSIL stage, and invasive cancer stage. This is the normal epithelium. This is the after the infection, certain period. Then the normal cell will convert into LSIL, low grade squamous intraepithelial lesion. This is the one. Low grade intraepithelial lesion. This is the one. After the infection, viral infection, the cell has been changed into this model. Isn't? After that, then HSIL, that is high grade. You can see the high grade is in the C, the high grade lesion. This cell converted into another cell model. We cannot come to the conclusion whether this is a cell or dust particle. It looks like a dust particle. This is, at least we can accept, this is a cell structure model. You can see here, this is the another one model. This is called high grade the squamous intraepithelial lesion. What is the lesion? In pathology, medical term, they are calling any changes in the cell we call lesion. So this is a low grade lesion. This is high grade lesion. Another one is atypical squamous cell in undetermined significance. This stage, we cannot say this is a lesion, so we cannot ignore it. This is called undetermined significance. And another one is ASCH, USC. We call USC. What is this? This is atypical squamous cell, but we cannot rule out whether it's a high grade or low grade. We could not able to rule out. That's why this is undetermined cases. These two are based on this. We cannot come to the conclusion. We could not pretty sure whether this lesion infected by HPV, like that. Another one is atypical glandular cell. You can see these cells are aggregated. It's like a magnetic, some iron dust particle aggregate on the iron aggregate on the magnetic area. This is called magnetic bead aggregation model. This is called GC, atypical glandular cell, like that. In the normal epithelia, it looks like this is the normal epithelium. You can see this is the normal epithelium, isn't it? This is also normal epithelium. This is LSIL, and this is ascus. Is an atypical squamous cell. Undetermined conditions are there. So, based on the viral infection, our cervical tissue, the structure will be changed. So based on this structure will be changed, and finally what will happen? Our cell will, the cervix cell will convert into cancer. This cancer will be there in the particular area. You can see that particular. Then what will happen? It is spread from this area to all over the body. That is called the invasive cancer. They invert other normal tissue. For example, this is the healthy epithelium, is in the squamous epithelium. Then slowly changes. One cell become cancerous. Then this cancerous cell will induce the neighbor cell become cancerous, and from there they may enter into other area. Is it possible to collect the cancer cell? Yes, possible, freely available. If you scratch this one, you can get it. Or sometime if the cancer started, some cervix area, some patients, they may have the mucus. It's like our cold mucus model. Some mucus secretion will be there. That secretion, we can collect the free cell. Which cell? The cancer cell. This is the free cancer cell. Or maybe you can get it the extracellular vesicle, you can take it. Or cell-free DNA, cell-free RNA, all will be present here. When we scratch it, you can get it. So you can collect this material, or you can collect the patient blood to detect the viral antigen, like that. So this is the cervical cancer development. But though it is there, what is the progress is that this is the normal cell. As I told you in the previous slide, this is the normal cervical tissue of the healthy woman, isn't it? Then what will happen if the viral particle infect this normal cell? Then it will be progress to the cancer. This is called the CIN 1, 2, 3, like that. This is called the cervical intraepithelial neoplasm. Neoplasm means cancer formation. So this is the cervical tissue, epithelial tissue. Within the epithelial tissue, epithelial cell, the neoplasm, cancer formation will be there. This is within the epithelium. That's why cervical intraepithelium neoplasia, isn't it? So then the progression will be graded. Grade 1, grade 2, grade 3, like that, it will be graded. So finally, what will happen? This is the grade 3. It's a higher level, severe dysplasia, is an or carcinoma. Carcinoma means there's a cancer. This cancer not only present here, it will be invert other area, tissue, or enter into the blood and spread all over the body. Or maybe it will be enter into the lumbar region. It will be enter into the other area, neural area, or maybe it will be enter in the bone marrow. We don't know. So where it will progress, we don't know. Mostly cervical area, it will be there. isn't it? So this is the one. How does the virus causes cancer? This is the $1 million question, isn't it? We have, by nature, we have to encounter the viral cancer progression every day. Say healthy, we are all healthy, you are all healthy. So how we tackle the cancer, whether in our healthy body the cancer cell will progress? Yes, sure. Pretty sure our body will face a cancer cell, but our immune system, our natural protective mechanism will prevent the cancer formation. Best example, we have two proteins, P53 and PRB, two proteins, the protein 53 and then protein retinoblastoma protein. These two are the tumor suppressor protein. Any tumor formation, these two proteins stop the cancer formation. Then these two protein will give the alarm to immune cells. The immune cells will come and destroy the cancer cells in the form of apoptosis. So here this is the mechanism. What will happen, as I told you before, in the human papilloma virus, E6 and E7 protein, early protein, these two protein integrate with P53 and go for ubiquitin conjugative enzyme complex and degrade the P53 protein. This P53 protein is like a security guard. They act as a security guard to prevent the cancer formation in the normal healthy people. But if the viral infected, especially high risk virus infected woman, the cervical tissue, what will happen? The viral E6, this protein will integrate with the P53 and degrade the P53. E7 integrate with the protein RB, that is called retinoblastoma protein. That will also be inactivated. Both will be inactivated. So cancer, that there is no protein will prevent the cancer formation, no barrier at all. So what will happen? The cancer will develop. So by this model, the viral protein prevent the tumor suppressor protein. Naturally, we have tumor suppressor protein. That protein will be suppressed by the viral protein. That is the main issues. So this is the main one, and the viral progression and how the cancer form like that. So, but anyhow, in UAE, the very limited number of studies are available. In 2023, we have done one study by using reverse hybridization method. Same method, but the methodology is a different methodology. Now we have changed it to real-time PCR. So we need more detailed study on the prevalence of human papilloma viral genotypes in this existing population, and as well as how many floating populations are there, whether they have HPV with different genotypes or same genotypes like that. So the study aimed to, the current study, what I am presenting now, this study aimed to explore the prevalence of HPV infection and its genotypes among the women with a normal and abnormal uterine cervix, whether it's when we see by using a normal eye or a coloscope or other thing, the cervix will be the normal, but it looks like normal, isn't it? Maybe the cervix will be infected by HPV through the sexual mode, or clear symptoms will be there. That is called the cervicitis will be there. So we have to know whether the normal cervix, uterine cervix, with a normal or abnormal. So what are the HPV genotypes are there? So first we should get the approval. Here UAE, if you want to go for any studies related to the human population or human-based studies, so we should get the ethical approval. So our university is provided. We got the ethical approval from the ministry, and as well as we got the approval from the university. So this study we have to design as per the World Medical Association WMA declaration guidelines. So we cannot design as per the normal one. As per the World Medical Association, what they have given the guidelines, we should follow the guidelines. Otherwise, it will be very difficult to face the further problems. And then along with that, the researcher, those who are doing research, we should get the certificate, good clinical practice certificate. As per the guidelines 2023, we should follow the guidelines. Then only we can start the research work. So our research work started nearly 1 year study a cross-sectional study conducted on the liquid-based cervical cytology. What is a liquid-based cervical cytology? This is the brush. This brush is called a cervarix. We insert into the gynecologist, not the study group. So we request the gynecologist. The gynecologist will use the brush and go for the scratches, and that means a scrape. Just a scrape. It's a very smooth brush. This brush, nothing will happen. This brush never tear the tissue. Okay, just scrape, and then the free floating cells, cervical cells, will be collected on the brush and keep it on the sample tube. This tube is contain 10 ml to 20 ml, mostly 10 ml formalin mixed, 5% formalin mixed buffer solution is there. This solution maintain the cell viability, prevent the cell death. Which cell? That is the cervical cell. So this is called LBCC, liquid based cervical cytology sample. We collected from 24 to 25 Jan. We collected almost all the Emirates, that means states of the United Arab Emirates. Once the sample we collected, send it to the Department of Pathology and Microbiology. We have our hospital laboratory that is called the Tumbe laboratory. And then the lab is there in the Tumbe University Hospital in our along with our medical university. So the study populations, we selected the study population age between 20 to above 55, but up to 55 we have keep it, because after that post-menopause the sexual activity will be less. So 20 to 55 years we have selected the women. Those who are attending the outpatient department of OBG and the gynecology, our hospital, other hospitals across almost one third of the United Arab Emirates hospitals. So except two major cities, we could not able to reach there. So all the samples we collected from the gynecologist. Actually, originally the gynecologist collected the samples, and then once the sample collected, keep it in the sample box and send it to the laboratory for cytological examination. This is the picture how they do, but not this brush. This is the very soft brush. So very soft one. We won't feel anything like that. This is just for explanation. This is a oral collection. Even the HPV viral particle, we can collect it from the oral in the buccal area. So for that purpose, we are using this brush. After that, we can go for the staining. You know very well about the staining, gram staining, other staining. Same model. We have to go for the stain. That stain name is called Papilloma stain. So this is the person, Dr. George Papilloma, is the person, he used this staining technique we call Papilloma. That's why the name came Pap smear. So Papilloma smear, like that, Pap smear. So this is after the staining, then the slide should be examined by the qualified pathologist. As a microbiologist or immunologist or anatomist cannot identify the slide. Only the qualified pathologist will examine the slide and give the report. You can see here, this is based on the slide, based on the Bethesda system. That is this is the Bible for reporting cervical cytology. As per the guidelines, so the slide will be examined and then identified. Then once the cytological examination over, remaining sample, we have almost 10 ml samples we have. Remaining 8 ml, we can go for the DNA extraction, HPV DNA extraction. So sample by magnetic bead method. So lysis the cell and then lysis the cell. Can you hear me? Hello?
Yes, sir. Yes, sir. Audible, sir.
Okay, ma'am. Okay. So here you can see the DNA extraction from the cervical sample. Cervical sample by lysis method, magnetic bead attraction with the DNA, and then wash, remove the debris, and then bead and the DNA will be attached with the magnetic bead with the magnet, and then finally elute only elute the DNA from the bead, and then we have to separate the bead. So this is the crude sample after the lysis by using the lysis buffer, DNA extraction lysis buffer. Then go for the multiple wash and remove the viral proteins. Only we need the viral DNA. And even the cervical cancer DNA, cervical tissue DNA is there. That will not affect our this one PCR work. So once the DNA extracted, including the cervical tissue DNA is there, nothing will happen because we are going for another step, PCR. This is RT-PCR, real-time PCR. What will happen? We have primers for all the 32 genotypes. We have the primer. The primer attached with the quencher and then attached with the fluorescent dye with a different color dye. This color, this color, this color, this color. Different color dyes are there. We call FAM and Hex and ROX and CY5. So four different fluorescent dye we have attached. Already the kit has attachment. That means the real-time PCR kit that has the primer with fluorescent dye. So here, what is the main principle of real-time PCR is called peptide nucleic acid based fluorescence melting curve analysis. Actually, this is the DNA. The human papillomaviral DNA. This is the primer. The primer will attach. After that, what will happen? We have to multiply. So by polymerization. After the polymerization, we have to increase the temperature. What will happen? Certain temperature, certain tolerable temperature, the primer will detach and then fluorescent dye will disappear. This is the end point. This is the point. By using which temperature, what temperature, it's exactly detaching, disappearing. That is the detection of particular primer. That means particular genotypic primer. So here the sample preparation, we use the sample kit from the Korea. This is called PANA real typer PCR kit to identify 32 genotype kit. So almost 20 high risk and 12 low risk. What are the high risk? You can see these are all the 20 high risk genotype, 16, 18, 26 like that. So you can keep it in your mind, 53, I will discuss later. So this genotype we have identified almost most of the people. This is the one of the highest prevalent genotype in our study. So like that, but internationally 16, 18, 6, 11, you should know 16, 18, 6, 11. We will come to know the last one. So these are all the genotypes are some are important, but other genotypes also is there. So by using the reagent kit, like a Panagene real typer kit, one is called ABO, and then this is the fluorescent dye. This is another fluorescent dye. This is another fluorescent dye. Like that, they have given 3 different fluorescent dye with the Taq polymerase, positive control, negative control, all are there. The overall reaction volume will be 25 microliter. This is the PCR protocol, pre-incubation and then initial denaturation, annealing, extension, maybe undergraduate student, like a nursing student, and then biochemistry, undergraduate, postgraduate, even MPhil or PhD student. You know very well about the PCR. I hope so. I think so. So this is the protocol for the PCR protocol. But which machine we can use it? The PCR, real-time PCR for this kind of the Panagene sampling process, that Panagene that is called genotyping kit, we need the special machine. Any one of the machine, maybe Biorad or Thermo Fisher. We have Biorad machine, this machine, CFX 96 real-time PCR detection we have. So we use this one. It's compatible with the reaction kit with the reaction cycle. And then in this kit, I told you, so one is fluorescent dye FAM, Hex, and ROX, and Cy95, CY5. These fluorescent dye, they have mixed in the A tube, same one mixed in the B tube, O tube, but this is a different primer for 33, 11, 58, like that. Different primers we have designed with the tag with the fluorescent dye. For example, this primer, the heat acceptable temperature 45 to 49.5. So 49.5, 0.6, it will disappear. So 49.5 is the last tolerating temperature. So 33 like that, 60, 11 like that. So when we apply the heat, what will happen? The attached primer will be remaining same. One particular temperature will be disappearing, color will be disappearing. That is the catching temperature. That is the notable temperature. That is called the proper thermal timing. So that temperature will be the indirectly proportional to that particular genotypes. So for example, internal control also it is there. These are all the reaction will be there, but we should have the internal control. This internal control genome will be attaching with the primer. That primer will attach on the internal control at the temperature 65 to 73. Then the internal control will also be disappear. The color will also be disappear. Here we have to polymerize and attach the coloring agent. After applying the heat, the color will be disappear. This is the main principle. But the temperature will vary from every genotypic primers. That is the main one. So in the internal control detected and the target detected, that is positive. Internal control detected, but a target and the pap smear sample, there is no detection of HPV, that is also valid. But internal control not detected, but HPV detected also we can consider valid. But both internal control not detected, target also not detected, that means this reaction is invalid. We cannot give the report to the patient. Isn't it? This is the one melting curve. This is called melting curve. You can see the peak 1, 2, 3, 4. So here also 1, 2, 3, 4. Here also 1, 2. This is the positive control, because usually the melting curve, it will appear on the display. It's like a peak 1, 1, 2, 3, 4 like that. If this kind of peak will be appearing on the display, this reaction is positive. This kind of one is the positive. This O is for validity. Yes, internal control validity is there. So this is X, the blue color for X and yellow color for rocks, and then green color for blue color for FAM. X is called the green color, yellow color is the rock, and the violet color for cyanine 5. So these color variation easy to identify the fluorescent dye. Negative control, the reaction will be like that. So this is a negative. The reaction no peak. The peak is the inverted model, negative model. So negative, then you can see the O is peak is the this is valid. That means this is a negative control and the O is valid, acceptable. That means the person is negative to the HPV. So for example, sample 1, sample 2, sample 3, sample 4. Sample 1 collected from 45-year-old female. In that female, you can see the line is there. So FAM is the blue color positive, isn't it? B negative. And then O internal control positive. This blue color attached on the temperature, the type 11. That means she has genotype, HPV genotype 11 positive. And then sample 2, you can see this is A negative, B 1, 2 is there. So 2 peak is happen. One is FAM, another one is cyanine 5. That means 2 genotypes, 51, 35 is there. O is the internal control validity. So same sample 3 like that, sample 4, sample 3, 18 and this is 1, 18 orange color, and as well as 35 is there. Yeah, 35 is there. Like that, this is the sample. Here you can see the sample 4, no peak line, A and B. O is internal control positive. That means O is positive, internal control, but the sample is a negative. So HPV not detected. That means she is no HPV infection, negative result like that. So this is the one. Sorry. Yeah. What is the result part? Yeah, here the part is that in our study sample, we have collected 229 cervical smear sample we have collected during the study period. And then all the women samples we have grouped into the age group 20 to 30 years old female, 31 to 40 year female, 41 to 50, and then above 50 like that. Because above 51, the sample size is very limited, 8. But here 41 to 50, 48 patients, 31 to 40, 96 patients, 20 to 30, 77 patients. So total 229 patients are there. According to the cytological examination, this is the lesion, ASCUS, LSIL, AGC, ASC like that. But NILM, normal epithelium. That means we could not identify any different lesions, any changes. This is the normal epithelium. 167 patient sample is the normal epithelium. But among this normal epithelium, when there is no cytological changes, but 49 out of 167, 49, they are truly positive, but no symptom, nothing, anything. So no symptoms at all. They are like a normal, but they have HPV. Like that, this is the tabular column we have prepared. And then this is the real patient sample from our studies. We collected the sample and examined the sample, and we have shown here. We are presenting here. This is called LSIL. This is ASCUS. This is AGC. This is ASC, like that. So such kind of changes happen. Then we can predict. We can come to the conclusion most probably HPV. But here you can see the HPV is a negative, but LSIL. This is LSIL, but when you go for PCR negative, most probably the different lesions are not only caused by human papilloma virus, maybe some protozoa, Trichomonas vaginalis, that will also be transmitted through sexual mode, especially for young couple, and some other sexually transmitted bacteria will also cause the disease. So sometimes possibilities are there. Mostly HPV related lesions are there. Sometimes other than the HPV, sexually transmitted pathogen will also causes the cytological changes. So you cannot rely on the cytological changes. We have to go for the molecular diagnosis by using PCR method. So in our studies, you can see how many people, almost nearly more than 70% of the people, they have normal epithelium, but infected. And then ASCUS very less, and then LSIL very less, and ASC is very less. So this is the age group of the study population based on the cytological changes. By using these cytological changes, we can come to the conclusion most probably those patients may be infected by HPV. Then among that, in that population, how many are HPV positive cytological changes, negative like that. Here, one beautiful finding is there. So we have grouped the patients, so we have grouped the study sample, study sample 20 to 30, 31 to 40, 41 to 50, above 51, all are there. So that age group, again, we divided into two groups. One is Arab study population, non-Arab study population, ethnicity wise. Arab ethnicity, non-Arab ethnicity. The people from any countries that is belongs to Arab, and any people, those who are from any countries, and that is a non-Arab ethnicity. So HPV positive, negative. What are the cytological changes? Whether the the positive person, they have single genotype, yes. High risk, yes. Or low risk, yes. Low risk, isn't it? And multiple HR, high risk multiple, and mixed low risk, maybe more than 2, 3 like that. Mixed H and LR, that means some patients, they have more than 3, 4 genotypes, and from a single sample. You can see 1, 2, 3, 4, 5, 6. 6 genotypes present in together in one patient sample. I will show in another one is there. You can see here, this is the patient. Here you can see this patient. This is a non-Arab study population. The patient, we don't want to disclose the patient and other things. So belongs to the non-Arab. So she has 1, 2, 3, 4, 5, 6, 7, 8 genotypes are there. Here also 9 genotypes are there. You can see 9 genotypes. At such a single sample, these many genotypes are there. So sometimes the patient may have single or double or triple or multiple, maybe all are same high risk, all are same low risk, or mixed together like that. So this is the one. You can see below after 50, above 50, 50, above 50, you can see very few genotypes are there. So this is the tabular column. So this is the graphical representation, frequency of uh, uh, we will do it now. So this is the graphical representation about this one. What are the genotypes are present, and this is the frequency. Here we can see the graph in the 229. So there is this Arab population. This is complete Arab, Middle East and North Africa. This is Asian country. Among the Asian country, Thailand populations are more, and here UAE other populations are there. So in our studies, the Thailand population, they have infected nearly 40% of the people they have infected. And here only 15-16% people are infected. Other country populations are infected. So this is the frequency we have identified. Isn't this is overall the frequency, isn't so how many people at the age of 20 to 30, 31 to 40, 41 to 50 above, isn't but this is the number of single genotype, multiple genotype mixed all together based on the cervical lesion, cervical cytological changes with the graph model. This is called a bubble plot like that. Okay, so that's the one. Why I'm telling here, almost this is the main one. Many studies they have mentioned that, isn't it? So here the main highlight is there, isn't it? So nearly low risk genotype is the 26% with a single and multiple other. But main identify one is what is this? I will let you know. This is the one. HPV-53 is the highest one, and the next one is 16. And these are the genotypes are there. So in our studies, we have identified 53, 16, 68, 31, 66. So even in the Arab study population, even in the non-Arab study population. So Arab study population 53 is the highest one. In the non-Arab population, 66 is the highest one. Why I'm telling this is the one. So we are detecting other genotypes are there. Yeah. So what is the main reason? Actually, this is the thing. You can see as of now, we have the vaccine for human papilloma virus. We have the vaccine. This vaccine is called a 9-valent vaccine. In other countries, bivalent vaccine. 2 bivalent vaccine is there. Another one is a tetravalent vaccine is there. Bivalent vaccine, and then 16 and 18 is the bivalent vaccine. Tetravalent 6 and 9, 11 is the tetravalent vaccine. But you can see 9-valent vaccines also available. Those vaccine could not able to cover these genotypes. These genotypes could not able to cover. Few are covering, few are not covering. You cannot see. This is the main one. Most of the genotypes could not able to cover. So that means, so we don't have the solution for other than these 9 vaccine genotypes. So we need more further studies to develop the vaccine designing according to not only for the international, according to the regional. For example, certain genotypes present in the particular continent, particular country, some certain genotypes is present in the many countries. So like UAE, it's like other Western countries. This is the international populations are living. So many people they come from many countries and residing here and go back. This is like a floating population. So such kind of floating population, we have to consider. We should think whether we can use the existing vaccination or we can go for the vaccine designing. That's it. So this is my study. I hope you have understood something like that. If you have any doubts or clarification, please you can discuss with me. Okay. Yeah. I think so. I hope so. The students maybe might be understand. Any doubts or anything, any clarification, please you can ask me, especially nursing students and biochemistry students. So you may work on this field, maybe in the molecular biology, or maybe the nursing students for the diagnostic side, especially for gynecology department, like that. Yes, ma'am. That's it. So now I stop my presentation.
Good afternoon, sir.
Yes, good
Sir,
afternoon, please. Yeah, please.
sir, if any unique or uncommon HPV stains in Indian samples.
Yeah, we found actually, actually now based on the patient flow, we can check it. Here we have some limitation in our study. We cannot ask the patient, you can participate for our study. When I ask the patient, can you participate in our study? We should get the government permission. We have to go for the ethical clearance. If any damage happened while collecting the sample, anything goes wrong, we are the responsible. And without their knowledge, we cannot use their study samples like that. Here we have a lot of limitations happen. This is the pilot study. We started from 2-3 studies we started. This is the pilot study. So ministry approved our studies. Now we have full-fledged, we are working. Now again, we are continuing. So we are continuing the studies. But one thing is there, Indian sample is there. But now the India has promoting the vaccination, especially Tamil Nadu government also promoting the vaccination. I have seen the human papilloma viral vaccination. That is good actually. Not only for male and even not only for female, even male also they can take it. That will be very good, isn't it? But especially for female, because it's a silent and invasive, it will be present there and then try to cause the cancer, isn't it? But we have to check it not only for single study, we have to continue for 5 years whether the same strain, same genotypes present in the same study population, particular nationality. Then we can come to the solid conclusion. Last 5 years, the same genotypes present in the, for example, Indian sample or Bangladesh or Asian or Thailand or maybe Middle East people. Then we have to go for the ministry information to design the vaccine, to include the vaccine, as well as the biochemistry students. You have the molecular biology, PhD, MPhil, PhD, even the bioinformatics, you can use the tool to design the vaccine. I think you catch my point. Yes. Any questions?
Good afternoon, sir. My doubt
Good
is, the vaccination rate influence the HPV genotypes?
Pardon?
Whether the vaccination rates influence the HPV genotypes?
No, actually the vaccine reduced the one mainly, actually, this is from the history. They have developed the two vaccines, 16 and 18, they have identified. Later they have identified 4, 16, 18, 6 and 11. Now they have reached up to 9 vaccine. But those 9 vaccine could not able to cover all the existing genotypes. Sometime one genotype will be present one year, within less than 1 year. After that, it will be disappearing. So it's like a rhythm. When it will be coming, when it will be disappearing, we don't know. So we have to go for solid studies. Now the WHO, they have funded for the society, Human Papilloma Viral Society. Just last month, October, they have conducted the conference in Thailand. So last time we had a meeting with your management and principal ma'am, nursing principal ma'am. So if you nursing students, you have to go for masters, especially gynecology, then go for the other things, even the biochemistry also. But the HPV based on the vaccination, it will not be increasing. It's exploring. Actually, we don't have the facility to unexplore. Many things are there. We don't have the exploration. Now we are exploring. Now we are studying. So many fields are there, unexploring fields are there. So based on the vaccine, it's not progressing. So now we are initiating, we are going for the development of 9 vaccine. Last time, 1.5 months before we had a conference in the infectious disease, I met our Serum Institute, Atar Poonawala, from our Indian Serum Institute. All of you know about this. This institute famous for the COVID vaccine. They have invested more than $400 million money for the vaccine development. Now they are developing the 9 vaccine. So they have 4 vaccines are available, and the 9 vaccines also now they are developing. Maybe within less than a year, you will avail the 9 vaccine. Like that. So what is the difference between here and the India? But India, there is no other country populations. We are sustained there. So any viruses are circulating within the Indian only, it will be circulating. But here it will be different. So this 2-3 years, the people will be coming from other countries. They can spread and then they can move. So like that. That's it.
Shall we end the session, sir?
So, good afternoon, sir. So, very nice, interesting session. So, I have one doubt. As you said that we cannot identify some of the women infected with.
Can you speak loudly, please?
Yes, sir. So, as you said that many of the women are not identified, but they have the infection. Then, how could we identify earlier to prevent the occurrence?
Yeah. That's the thing. Actually, what is the reason? We have to go for more awareness. Even we have complete awareness program. So, like in the nursing college, I saw in the site they have a lot of awareness program. Here also we are conducting the awareness program. Sometimes people they are not here. They can go for vacation. They can go to the country and come back. Some of the people are in the low income. People are there. So they have hesitate. I don't have anything. I am normal. So why I should go and waste my money like insurance, like that. This is the ignorance from the patient side. The people side is that. So now we are promoting more. The government has the step in 2030. Almost 96 population should be vaccinated like that. Still the government promoting, but the people ignorance are there. That is the main one is that. So I cannot say my work is 100% reachable. This is the pilot study. A lot of limitations are there. We don't know many patients whether vaccinated or not vaccinated. A lot of students we interviewed in our university, medical university. You got it? Have you got the HPV vaccine? Yes, I know, I got it. So what type of genotypes? We don't know, like that. Because when they are in the school, they have given the vaccine, but they don't know which type of vaccine they have taken. We come to know, yes, most probably bivalent or tetravalent, that's it. So recently 9-valent they have included. So based on that assumption, we can come to the conclusion. So this should be, we have to, in our studies, we have to rectify that in future. Now we are continuing the work.
Yes, sir. Thank you so much, sir.
So yes, ma'am. That's it.
Shall we end the session, sir?
Yes, please. Yeah, if any doubts, please, no problem. So today is my holiday, no problem. Today is here UAE National Day, so I am free, no problem.
Thank you so much, sir. You have nicely explained everything. Like, I think they are the undergraduate students. They are
Yes,
learning
yes, ma'am.
stage. Once they come to the research level, maybe your session may be more helpful for them to utilize whatever the inputs you have given. Both biochemistry students as well as the nursing students.
Yes, ma'am.
But
Actually,
clearly all the pictures, everything nicely have noted and familiarized to the
ma'am.
students.
This
It
is
is
the
so nice.
undergraduate students. This is for just ignite their curiosity
Yes, sir.
to focus on the field. Because my dear students, I have an advice: don't be like a bookish one. You have the social platform. So don't waste your time. Don't waste your money. You can use it for many useful activities like that. For example, for young students, you are the pillar of our nation. You should go for activity. You should keep it in your mind, achievement. So you should be that is called a no pain, no gain. You should be hard work, and as well as for young females, lot of societies, international societies, they are offering, they are giving the free the membership. You can be a member. For example, biochemistry society member. You can go for conference. You can go for presentation, poster presentation. You can start a lot of small ideas. There's the survey in Virupuram district survey. Many things you can do, and then you can have the idea for the masters and other thing. So like that, many countries they are helping. So especially for our Indian students, many countries now the German they have opened for postgraduate with the government fees, not in the international fees. And many universities they are giving the scholarship as well. So now the platform, social media platform, is to globalize everything within in our hand. So if you use that platform in a right way, you will be achievable.
Yes, sir. Definitely,
Yes,
sir.
yes, ma'am. Thank
Actually,
you.
nursing is like evidence-based practice. It is focused and expected only through research. We have to go for all the treatment. As well as they are the backbone or allied courses. They are supporting for us to take the like a care to the society through us.
Yes.
So, thank you so much, sir.
Thank you,
Nice.
ma'am. Thank you very much. Thank you, ma'am.